is Mr. Maxwell for Pingry AP Biology. We're in the middle of the information unit, and I think people are feeling a little bit like we are a little bit disjointed, and some of that's actually true because of the discontinuities caused by me having surgery scheduled and then canceled, and then having to go remote and be in person. So I wanted to stop and give you a little bit of, to tie these things together a little bit for you, because I'm feeling like they're a little bit disjointed too. So first off, I've listed some topics here that we've covered so far, and then things that we have yet to cover. Now, we started out with DNA structure and how we know DNA is the genetic material, and we did a little bit about replication, and so you need to have that information so that you can understand how genes are expressed. Genes are located on DNA, and we did the gene hunting activity where you looked for a gene, found the promoter, found the terminator, and then transcribed it, and then translated it into protein. And then we kind of went back and looked at genomes, which are, a genome is a collection of all of the genes in a particular organism, and how single nucleotide changes in those genes in the genome lead to different traits. Okay, now traits historically are what we look at when we look at inheritance. To understand inheritance, you have to understand how cells divide. So that's where the cell cycle comes in, because the cell cycle is what allows you to copy DNA and then to make a new cell. Mitosis is the process where in eukaryotes you divide those genetic contents up into new nuclei so that you can make new cells. And then meiosis is the sexual version of that, where instead of making identical asexual nuclei, you make sexual nuclei that are haploid, and then you stick them back together to make a diploid nucleus. And the diploid nucleus, we're diploids, and so you have to do meiosis to reduce the number of chromosomes, and then do fertilization to bring it back up to the full number. And so the genes are moved around in that process. We also started an experiment with sea ferns where we will look at how inheritance of a trait is sorted in the sea ferns. The trait that we're going to look at is a polka dot phenotype, and we'll be able to see that result of meiosis from an F1 that is heterozygote, and so you should get a 1-to-1 ratio in that. And sea ferns is how we're investigating inheritance. Often we also do fruit flies, but I don't think we're going to be able to do fruit flies this year. But sea ferns, we figured out how to be able to do them at home. So the sea ferns the spores that you inoculated will grow by mitosis into multicellular haploid individuals, and you'll be able to see, should be able to see, the 1-to-1 ratio that you're going to get out of that. And then the sperm that those produce will mix and produce diploids. Most of the inheritance studies, most of the inheritance information you're going to see is going to be based on, like humans, where we go diploid, briefly haploid, and then back to diploid again. And so some of the problems that I've given you are the simple Mendelian monohybrids. And then we're going to add on top of that chi-square testing. So when you do an experiment where you get offspring, the offspring typically are not going to be a perfect 3-to-1 ratio or 1-to-1 ratio or 1-to-1-to-1 ratio. They're going to be a little bit different. And so researchers use chi-square to be able to tell, are the data that I got equal to the ratio that I expected or close enough to the ratio I expected, or are they different enough that I have to throw out my hypothesis that it's not a one-to-one ratio and something else is going on. And you're going to see with the sea ferns both situations most likely. So we're going to use chi-square to analyze the data that you get from sea ferns. Okay, now a couple of topics that we haven't covered yet include signal transduction, which is how cells respond to external signals and then make internal changes in them. And that signal transduction can lead to a response such as contraction of muscles or a nerve impulse being fired, or to control gene expression, to turn on, to turn on the production of a particular protein. And so these things are all information, the way information moves in cells, and in organisms and in environments as well. And so that's the organismal level. And then we're also going to look at other responses to external signals, such as pheromones. We'll talk about pheromones a little bit. So organisms like fruit flies will do like mating dances, and those are also external signals that they have internal responses to. And so we'll talk about those types of things too. I hope that helps to kind of gel things together. The central unit of the central theme of this unit is information, and all these things are ways that information is used in biological systems to make things that are more complex. So.